Today we're going to teach you all about EEG, also known as electroencephalography.
Well, EEG is a piece of equipment that records electrical activity in the brain through electrodes placed on the scalp. The electrical activity is presented in the form of brain waves on a polygraph.
What is a brainwave?
A brainwave is an electrical impulse in the brain, and researchers can look further into these brainwaves through the event-related potential technique, also known as ERP.
ERP?
ERP can show a timeline-like recording of brain processing through the high temporal resolution. High temporal resolution means we can view the brain processes and responses in real time. EEG could be seen as the least invasive method of assessing brain activity, so we don't have to worry about any negative impacts on the participant.
So what are some other advantages of using ERP?
Well, although there are many positive aspects of using EEG and the ERP technique, other methods of measuring brain activity, such as fMRI, can see where the processes and responses are taking place in the brain. EEG cannot do this because of the low spatial resolution it holds. However, this should not take away from the great temporal resolution that allows us to assess and identify specific brain waves that are linked to certain processes.
Oh, it sounds great. So we have different brainwaves for different processes?
A particular brainwave that has been linked to many processes is the P3 component. The P3 is a large positive brainwave which is located in the parietal lobe of the brain, which occurs around 300 to 400 milliseconds after stimuli onset. Researchers have associated the P3 component with semantic processing tasks with meaning and emotion responses. This has been found through using the oddball task. There's a large amount of research based on EEG and the concept of relevance, and it has provided a vast amount of knowledge about brain processes. As research is advancing, our understanding of EEG and its components such as ERP and P3 becomes more clear. Multiple studies have identified that the P300 wave tends to peak at 300 milliseconds. Also, the P300 spikes when an individual identifies something important or relevant compared to something irrelevant. This has been identified in multiple studies, such as Donchin 2000. This was actually one of the first studies created about this specific topic. This study focuses on assessing P300-based brain-computer interface. Participants have to focus and view a matrix, which is where characters are presented. They must observe and count the number of times the row or column containing the designated target letter P was intensified. The result of this study found that it is possible to construct a brain-computer interface that, using P300, allows an individual to operate a virtual keyboard without using or requiring any movement of the body, as in the skeletal muscles. This study has contributed greatly to the knowledge of brain processing, as it identifies that EEG and P300 accurately identify the different components of the brain and emphasises the concept of relevance.
Another study by Sutton et al. 1965 looked at evoked potential correlates of stimuli and certity. They averaged evoked potential waveforms to sound and light stimuli recorded from the scalp. This showed differences between reactions such as light and sound. Reactions tended to be longer when the participant was certain, meaning that they knew/expected the stimuli. This study identifies the importance of stimuli to the subject and that different stimuli can create different reactions. This relates to the oddball paradigm, as it displays different response will be generated depending on the familiarity of stimuli.
We aim to get similar findings from a similar relevant stimuli task we will conduct. Example of the oddball task. So, the rapid visual presentation task is an example of a measure that uses the oddball paradigm to access the P3. Here is an example of the task we have created. First, we will walk you through the task step by step and explain the use of relevant, irrelevant and oddball stimuli that is used throughout. We will then run through it as you are taking part in the task.
Okay.
So the task will begin by welcoming the participant and asking to press any key to begin the task to ensure they are ready to proceed.
Okay, so shall I press any key?
Yeah, sure.
Okay.
So then, the participant will be shown all the images that will be presented throughout the task without information on which is relevant or irrelevant. They will be asked to identify their own image by entering the number beside their image in the box on the bottom right.
Just there?
Yeah.
Okay.
So, the participant will then be presented with the image to keep in mind. The image will be any image displayed on the previous slide, aside from the image of pets, which will be the oddball. This will be identified as a relevant image of the first section of the task for both the participant and the researcher.
Okay.
Once given an image to keep in mind, the participant will be asked to identify which number correlates to the image they have been asked to remember. This ensures that they've paid attention to the relevant stimuli, ensuring the researcher can access when the participant is presented with their relevant image. This process of having a relevant image to remember will be repeated with each image, aside from the oddball pet images.
Okay, so this one would be number 2.
Yeah.
Okay.
This image is irrelevant as the participant has not been instructed to keep this in mind. This will be part of the irrelevant relevant series of the experiment, along with the remaining irrelevant image.
Oh, that's me.
The image should spike a P3 wave as it is relevant to the participant at this time. This image ends the first of the 2 irrelevant relevant series of the experiment. So, this image begins the irrelevant relevant odd series of the experiment where all irrelevant images are presented, along with the relevant image of the participant has been asked to keep in mind and the odd image of a pet. So this image is the oddball of the experiment. It is neither relevant or irrelevant and may spike a brainwave on the polygraph due to the image being odd to the participant.
Oh, okay.
This image begins the final irrelevant relevant series of the experiment, whereby the odd image is now taken out and the participant is presented with remaining irrelevant images and the image asked to keep in mind.
Okay. Okay, so that's the relevant image again.
Yeah.
And the final irrelevant image.
Yeah.
Okay.
So to end the experiment, the participant will once again be asked to identify the image they were asked to keep in mind. This is to ensure they were focusing on the correct relevant image for the researcher to analyse the data accurately.
Okay, so that would be number 2.
Correct. And then for the final, it would just be thanking them for the participation. Pressing any key to end. Done.
Cool. We'll now take you through a trial run of the experiment to show you how to construct your own rapid serial visual stimuli task. Welcome to the task. Which image is your own? Type the corresponding number to your own image. Please keep this image in mind throughout the experiment. Please state the corresponding number to the image you have been asked to keep in mind. The first trial will now begin. In the full version of this experiment, there will be 62 images per trial. Of the first 24 images, the relevant image will be shown once per 6 images. Of the next 7 images, your relevant image will be shown once, an oddball image will be shown once, accompanied by 5 irrelevant images. The first trial of 24 images, 1 relevant per 6 images, will be repeated. And then the second trial of 7 images, 1 relevant, 1 oddball and 5 irrelevant images will again be repeated. Overall, this means that you will be shown your relevant image 10 times, an oddball image twice, and irrelevant images 50 times. Each image should be displayed for 600 milliseconds with a 50 millisecond blank screen in between. When organising the stimuli, be sure to edit all images to 300 by 500 pixels. Ensure they all look roughly similar with just the head and shoulders on display. These details should also apply to the images of the pets.
Here is an EEG experiment step by step. First of all, the participant is seated in the room in front of a computer, and once the experiment is explained to them, the experimenter will measure their head for size. An EEG cap, which is best suited to them, is placed on their head and fastened. Electrodes are separated into two cables, which are attached to A and B ports on an amplifier, which is connected to the recording computer and stimulus computer. The amplifier is then turned on. Electrodes are then placed on the cap along with gel, which helps the current flow. When electrodes are being placed on the scalp, a blue light will be present if everything is okay. If the blue light flashes, the electrodes need to be repositioned until the flashing stops. Once all 64 electrodes are placed on the cap, and 2 electrodes, also known as auxiliary electrodes, are placed directly above and under the left eye, the experiment can begin. A few trials occur before the experiment, such as asking the participant to smile, frown, blink, and so on. This is to ensure the EEG channels are working correctly. To check the channels are definitely working, the electrode offset tab is checked, as this identifies whether any electrodes need replacing. The participant goes through one trial with the experimenter to ensure they understand what they have to do. As the participant does the experiment, data is recorded and transmitted onto the experimenter's computer. The experimenter can identify the different brainwaves being transmitted. As the experiment progresses, the researcher keeps a close eye on the results coming through. This is to make sure that the participant is still alert during the trials, which can be seen through alpha waves. Once all the trials are completed by the participant, the data will be saved and the electrodes will be carefully removed from the cap, and the participant will be able to leave. Everything then has to be thoroughly washed, as gel can cause a blockage when it is not cleared. All electrodes have to be washed too in order for it to work effectively the next time.
In order to analyse our results, MATLAB was used. MATLAB is a data analysis software. It enabled us to separate data from the experiment and also identify and visually see EEG activity, including the different data recorded from each specific electrode. MATLAB also enabled us to go from a raw, continuous dataset to an epoch dataset. Measuring ERPs allows us to interpret which part of the brain activity is related to our task-relevant stimulus and no external factors. The assumption of ERP research is that cognition relating to a certain stimulus is happening at a stable time, unlike noise, which occurs randomly when an individual thinks about anything else. Averaging stimuli will allow noise to be removed, so we are left with only relevant brain activity. Time-locking trials to stimulus onset can allow us to epoch the data and see how different types of stimuli can cause different types of brain activity. This is what we have aimed to do throughout our analysis. To begin, we imported our data through the EEG lab. From this, we could view our channels. E' was programmed to trigger 252 for irrelevant faces, 251 for the relevant face, and 250 for the oddball. The number 100 meant the face was someone else's, and 200 meant the face was their own. As well as showing us the response to the specific stimuli, this dataset can show which channels we should remove. As you can see from this dataset, the channel C5 looks particularly flat, therefore we removed this channel. Not only do bad channels need to be removed, high levels of noise also need to be removed, as this is not the brain activity we are interested in. This can be seen through a hairy look on the dataset, which can be caused by muscle activity. The next step is to remove data below 0.25 Hz, which allows us to filter data, removing low frequency. The data then has a much cleaner look, and once time locking, we are able to delve further into artifacts and remove these. Asking EEG Lab to reject data seems to be an effective way of doing this. The following dataset displays rejected data through marking them in the colour green. However, in our analysis, we changed the threshold in order to retain some data EEG Lab suggested we reject. We did this with the guidance of this graph. Once completing this process, we found that through calculation of our epochs that our dataset was made up of 17.8% of relevant stimuli. Upon focusing on single subject ERPs, we were able to delve deeper into the components that were displayed in different channels, and therefore different areas of the brain. We found parietal occipital areas were most active closer to onset of stimuli, and central and frontal were least active. This is apparent in the channels C1 and FP1 in comparison to that of P08 and P04. In order to identify the first positive peak, also known as P1, multiple channels were assessed closely, which allowed it to become apparent that P08 was in fact the channel where P1 was visible. This process was repeated to find the first negative peak, and again we found that P08 also held the first negative peak. The spike at the end of this plot simply shows the 600 millisecond display of stimulus ending and the next stimulus being shown. As you can see from the following plots, the lines representing the type of stimuli run a similar track and then begin to diverge. This may be due to the psychological processing stages. The separation begins generally around 300 milliseconds, and therefore this may be pinpointed as the time in which the brain recognizes the stimuli differs from one another and each has a different meaning. P1 tends to occur simply due to the process of light in the brain. These findings stated previously were simply a single subject EEG analysis, but now it's time for the grand average. We begin by formatting all channels into plots in order to assess each channel closely. Our P1 appeared to be visible in channel OZ, whereby the PET condition gave a P1 from 46 to 85 milliseconds after stimulus onset, maximal in the OZ channel at 7.23 microvolts. Later potentials are visible in channels such as P10. This peak occurs before the end of the stimulus display and has a microvoltage of 5.48 at 416 milliseconds after stimulus onset. Because this study is focusing on the component P3, we assessed all channels to identify where our P3 may be. Parietal and occipital areas seem to hold high positive peaks at around 300 milliseconds, but it seems that P8 shows what may be the P3 component. To analyse the P3 component deeper, topography plots can allow us to view an almost map-like figure of the scalp at during the ERP recordings. The topographies can show us where in the brain is more positively or negatively active during times throughout the different stimuli. For example, here we can see that the parietal areas are much more positive during the oddball stimulus than a relevant and relevant stimulus. This is particularly interesting to see that the brain during the oddball stimulus gave much more positive recordings at the time a P3 usually occurs, especially in the parietal areas, than the relevant and irrelevant stimulus conditions. Due to research suggestions that the P3 occurs when the brain pays attention to a certain stimulus, this could suggest that the oddball stimuli of the PET spikes a P3 component, as the participant is not expecting this image and therefore pays more attention to the image. The oddball stimulus continues to have a positive reading up until the end of the stimuli display, suggesting this has a longer lasting potential than the other types of stimuli. Through these topographies, it is interesting to note that the irrelevant and relevant stimuli does not differ all that much, therefore it could be suggested that relevance of stimuli does not affect ERPs that greatly. An explanation for this could be the difference in faces. In the relevant and irrelevant conditions, human faces are shown, whereas in the oddball condition, an animal face is shown. This may be a cause for the difference in conditions. This clip shows there are no great differences between the relevant and irrelevant conditions.
The aim of the study was to assess the influence a relevant, irrelevant and odd image has on the electrical activity in the brain. There was emphasis on the P3 component as previous research shows that oddball tasks similar to the experiment elicit a P3 response.
We replicated previous studies as their methods have been shown to be a reliable way of measuring how the brain responds to various stimuli that differs on relevance.
The oddball tasks uses an EEG to look at the ERPs generated with the aims of measuring the multiple brain responses to relevant, irrelevant and oddball stimuli. Our results have confirmed previous research such as Ferrari's 2010 study by identifying the oddball stimuli spikes a P3 brainwave.
Our main findings were differences between conditions. Unlike much previous research, our findings shown almost no differences between the relevant and irrelevant stimuli. However, the oddball stimuli spiked a P3 response. The oddball stimuli also had higher positive brain activity than the relevant and irrelevant conditions. This brain activity was mainly around the parietal and occipital areas of the brain. This could suggest a link between attention and the P3 component. When unexpectedly shown an odd image, the participant may pay more attention to that image than if they were expecting to see it. This attention may also be linked to the parietal and occipital areas of the brain. The lack of difference between irrelevant and relevant conditions may be explained through the use of human faces compared to animal faces in the oddball stimulus. Although these findings support previous studies, this study used a majority female sample, which Campanella et al. In 2004 previously identified that there could be gender differences in response to oddball tasks. This could affect the generalizability of findings.
Another aspect of the study that was not controlled was fatigue of the participant. Research by Boxem and colleagues in 2005 shown that fatigue can affect the response to a task similar to that of an oddball task. This should be controlled in further research.
Future research could also focus on the different types of stimuli, such as auditory, potentially somatosensation, to see if this elicits the same response.
What is the effect of seeing a rare or odd stimulus?
So rare or odd stimuli spikes a P3 response, which means the individual has an emotional response to the stimuli. This emotion could be confusion due to the unexpectancy of the rare or odd image.
Can P3 be identified through the use of stimuli being presented to multiple senses, such as auditory or visual?
Yes, so researchers found evidence for the P3 occurring in both visually semantic and auditory semantic stimuli, showing that the brainwave can be linked to multiple senses. This has been shown in Kochobi and Lang's 2001 study.
What is the effect of seeing oneself?
Once again, an emotional response seen through P3 will be elicited. However, this emotion may differ to that of the odd stimuli.
What is the effect of seeing oneself on the effects of relevance?
Due to an image of oneself holding relevance to an individual, ERP should show a P3 response.
Thank you.
No problem.
What is the effect of seeing rare or odd stimulus?
P3.
What is the effect of seeing oneself?
P3.
What is the effect of seeing oneself on the effect of relevance?
P3!